and you're on.
Okay, well, hello there, Balticon. My name is Eric Haig, and I'm a professor of biology at the University of Maryland. And I am really excited to tell you about some wacky stuff in the world of biology having to do with the subject of immortality and other things related to living and maintaining life in situations where you might not think you could do it otherwise. So I'm going to go in here and do a little screen share, and hopefully this is going to work. Okay, hopefully you're all seeing that. So the subject of immortality comes up a lot in science fiction, and I've just put here 7 novels that one website regards as among the 21 best science fiction novels that deal with immortality as a premise of the story. This is certainly not exhaustive, and your favorite may not be on here. I'll just mention that the favorite of this particular website was actually Gulliver's Travels, because it was deemed in one of its chapters to be dealing with immortal people, and maybe that was at least the first one. So here we have a major theme in science fiction, and what I want to do tonight is try to give you a sense of the reality of immortality, animals that might actually be close to immortal, and what we might as humans have to do if we wanted to try to learn from those animals and maybe become closer to immortal ourselves. So here we go, hold on. Okay, so the first thing I want to do is just give you a little background on really, if we're immortal, that implies there must be normal immortality. What is mortality? What do we mean when we say mortality? And where did mortality come from? So we are all descended from single-celled organisms, and single-celled organisms, they tend to divide, right, and become 2 single-celled organisms. And then those might divide again, and you get 4 single-celled organisms. Now, in this situation, this could go on forever, and the world could eventually become completely full of these single-celled organisms. But we know that doesn't happen. We know the world does not fill up with one kind of life. There is some kind of process that's opposing this division and growth. And that is a kind of mortality, but it's unpredictable. So for example, as this first cell divides into 2, one of the descendant cells might meet with predation, and some organism comes and ooh, it's gone. Now the other one is still around, and so it's in a position, I'm going to put my little laser pointer up here. So this one can go ahead and divide. But when it does, perhaps one of its offspring finds itself in an unusually hot and unpleasant location and just dies and falls to the bottom of the ocean if it's an algae. So although there was a lot of cell division going on here, there are processes that take out individual cells being consumed, being in a bad environment, so that on average, most organisms tend to have a fairly stable population. Even though there's a lot of growing going on, there's also a lot of dying, and they tend to be balanced. Okay, and importantly, it's not clear here at the beginning which of the descendants of this cell are going to die and when. So although there's mortality, it's not predictable mortality. So when does it get predictable? Well, it gets predictable when we become multicellular. And I'm gonna get into that in a second, but I just remembered we like to go on digressions a lot in academia, and I'm one of those people who likes digressions. So I'm gonna tell you a little story about the one case I know of, a single-celled organism where there is predictable mortality. And this has to do with the humble baker's yeast, Saccharomyces cerevisiae. If you're enjoying beer right now or bread, you can thank this organism. It's a little fungus. This cell divides like the other one did, but you'll notice there's this funny asymmetry. There's sort of a big mama cell and a little baby bud cell. And when they finally divide, one of them is bigger than the other. Now, if you were to track at every cell division who was the mama and who was the baby, and do that for a long time, this is what the diagram would sort of look like. The cell buds, the bud pops off, and the mama continues, makes another bud, bud pops off, mama continues. Keep doing that. And if you keep following the mama cell in every division, you see that they are in fact not actually immortal. So here's sort of cleaned up data from a paper that I found that basically measures how likely it is that the mother cell is still alive after it's gone through a certain number of generations of producing buds. And on average, a cell can only be the mother cell about 20 times in a row before it dies. And a few die sooner and a few die later. And to get a sense of what that actually looks like, these are some micrographs or close-up microscopic images of yeast. And here on the left, you'll see what nice, young, healthy cells with a mama cell and a baby cell look like. But after 15 generations, this mama cell here produced a daughter, D14, that could not separate. And then it tried to make one more bud and it just gave up and died. So this thing obviously looks old, right? So even yeast can get old if you track them in the right way. Okay, so with that kind of weird exception though, for the most part, I think we would say that mortality in single-celled organisms is not predictable. There's things that take out cells, but it's hard to know which ones are going to be killed. But mortality gets to be more predictable when you reach the world of multicellularity. And of course, that's the world we're in. So here's a hypothetical multicellular organism. It develops from a single-celled egg. It's been fertilized. It forms this multicellular hamburger-looking thing. It's got a top, it's got a bottom, it's got something in the middle. And then there's a few cells within that that are capable of doing this process we call meiosis, which is a special process of subdividing the genome into its paternal and maternal components, recombining them and producing in the end new combinations in eggs and sperm. So that's the process, part of the process of sex. These can then come together again in the other part of sex in fertilization to make a new genetic combination. Now notice that most of the body is not participating in this process, okay? And so that part of the body that doesn't make the sperm or the eggs, we call that the soma, the part of the body that doesn't participate directly in reproduction. So here's a real organism that my lab at Maryland works on. This is the little nematode C. elegans, and it's about 1 millimeter long and about 1/10 of a millimeter across. And it doesn't have that many cells. There's about a little less than 1,000 cells in the main soma part of the body. And then you'll see over here, there are these 2 cells at the beginning of the larval life of the worm that are going to give rise to all of the germ cells that can make sperm and eggs. This whole part in black, all these cell divisions that you see tracked out here over the life of the worm, those are all destined for the scrap heap. This is the part of the animal that is not going to go on, okay? So mortality for the worm then means the death of its soma. And if it's lucky, it left some descendants in terms of its offspring through its germ cells. Now, because it's Maryland in the summer of 2021, there's a lot of other death going on around us that you might have noticed. And I'm going to, if I can get my arrow, there we go. This is a video I took at the University of Maryland campus just a couple days ago. And see if I get the sound up, you can hear what's going on. So as you know, cicadas spend 17 years underground. They come out, if they're lucky, they find a mate. They lay some eggs if they're a female, and then they don't last too much longer. So this one is on its way out, and if we look down the sidewalk, we see a lot of its brethren who have gone before it to the other side, and a lot of cicada death happening right now. So death is all around us, and the organism that we think of, the cicada in this case, is dead. So that said, right, I've just stressed how the worm and the cicada, and of course us as humans, we don't live forever, and we know at a certain, maybe cicadas don't have consciousness of their impending demise, but they definitely die, and we know that we're all going to die. But that doesn't mean that all animals routinely die and predictably die. And so I wanted to share some examples of some animals that kind of go against this general expectation that multicellular animal life should have predictable lifespans. So one real classic example of this is the planarian worm. Some of you might have seen planarians when you were in junior high or high school laboratory courses. They're useful for all kinds of biological research, but one of the neatest things about them is you can slice them up and they don't die. So this planarian here is a diagram drawn by a really famous biologist named T.H. Morgan, who went on to become the father of genetics in Drosophila. But before he worked on fruit flies, he worked on planaria. So this experiment is now well over 100 years old. And he was amazed to find that when he cut the worm at these different places, that the pieces would grow back a head on the head side and a tail on the tail side. And this is his description of what he saw. A new head appears on the anterior or head end of all the pieces. A tail appears whenever a posterior cut edge is present. He notes here, interestingly, that the new head is about the same size as the old head, no matter where it starts growing. So it's sort of, I guess you could say, it loses its neck in the process of regeneration. But you know, if you think you're going to kill planarians by chopping them up into pieces, you'd be wrong. And certainly that's something that we can't do. Now, another worm that is, well, sorry, yeah, I'm going to get to that other worm later. This same worm, planarians, they have been studied more recently by much more modern techniques, and the phenomena that Morgan was examining have been now understood to some extent. We know that the regeneration ability of these worms really depends on these cells that you see diagrammed here in these black dots. These are stem cells called neoblasts, and they're spread all through the body of the worm. And so whenever you cut them, these neoblasts will rush to the cut site and rebuild the part of the body that's missing. They are undifferentiated cells that can become whatever they need to become. And importantly, these cells are actually constantly being used at a low level to rebuild the parts of the body away from the stem cell zone. So in other words, the planarian looks like a static organism, but it's actually constantly regenerating itself. It's like a treadmill. It keeps looking like the same worm, but actually the cells that are on the surface are not the same, and they turn over quite frequently. Quite recently, there was a lab that actually isolated the specific neoblast cells, and this is a little movie they made of what they look like live in a culture dish. And here the DNA in the nucleus is stained with a blue dye, and this green stuff here is a protein called T-span1 that is the distinguishing hallmark of these neoblasts. The cells that have T-span1 on their surface have this unique property of regenerating the whole organism. It's not quite clear what T-span1 does, but of course, maybe, maybe we want to find out. So planarians, they've been studied for a long time, but people are still learning new and fascinating things about them. Okay, now another worm that looks kind of similar to a planarian, but is actually very distantly related, is called an acial flatworm. Acials are really deeply diverging animals. They're distantly related to all other animals. They've been studied by my colleague at the University of Maryland, Alexa Bailey, a great person. I hope you get to meet her at some point. And they're interesting for a couple of reasons. First of all, you notice they have, so their head is up here. You'll notice that their tail, they have these little lobes. These lobes are not just bits of their butt. They are actually the beginnings of new little baby worms. So these worms will split themselves along these fission planes that you see here in dotted lines. And depending on the species, they might make one daughter worm that has the same orientation as the mama. It might make two babies that are split down the middle and also have the same orientation as the mama. Or they might develop with opposite polarity, so they get eyes on the back end and they crawl away from the head. And there's two different species that do that. So these animals have amazing asexual reproduction. They can just split themselves into bits and make more. Not surprisingly, then, they're also very tolerant of being sliced up. And a grad student named James Sykes that worked with Alexa did some really neat experiments where he tried to see how small he could cut the pieces of this worm and still have them live. And he got really small. So here's an example of a whole worm and what it might look like. In the diagram on the left here, you're seeing some of the tiniest pieces that James cut from this acial worm. And so if he cut this teeny tiny little rectangle here at spot A, this is what you get. You get a worm that regrows with a normal head and the little lobes, just like its mama worm. The one place that you could not get away with this is if you cut it exactly on the boundary between those two soon to be separate worms growing in opposite directions. So this is in the species that has a head on the back end and a main head going forward. If you get right on the boundary, you get at what they call the reversal zone, a confused piece of tissue that doesn't know where its head is. And so this is the one place where the worm would not be able to survive this kind of a surgery. But it's quite remarkable. We're getting down to pieces that are like just a millimeter or two across and they can still rebuild themselves. Okay, another animal that's famous for being able to regenerate is a relative of jellyfish called the hydra. This is like a freshwater sea anemone is basically what a hydra is. And they can reproduce sexually. They can also reproduce asexually, which is what this little bud is. This is a little baby hydra growing off the side of its mom. Or actually in this case, its dad, because its dad has testes. So you can take these and also cut them up. And if you do that, again, like the planarians and like the acials, they will grow back. So if you cut them down near the base, the head will grow a new foot, and the foot will grow a new head. So that's a pretty impressive ability to survive trauma. And if you look at more closely what's happening to the cells and how they move in response to injury, you discover that the cells are not actually static. They are always, even before you cut them, moving around the body from near the neck to the top and from near the neck down to the bottom. And it takes about weeks for cells that are born in this sort of stem cell zone to reach on this kind of cellular conveyor belt down to the bottom of the hydra, where there isn't very much movement. So what this means then is, like the planarian with the neoblasts, the hydra looks like it has a static body with tentacles and a trunk, but it's actually constantly flowing new cells into that body and rebuilding it and reshaping it from new cells all the time. So this, again, like with the other worms that we looked at, it correlates with the ability to regenerate. If they're constantly rebuilding their body, it's not that big of a deal to replace a piece that's missing, because those cells were going to be in that fate before too long anyway. Okay, so to summarize then, there are invertebrate animals that are said to be experiencing negligible mortality. Now, that doesn't mean something might not come and just eat the whole animal and digest it, right? That happens. So that, again, that's why the world is not full of planarians and hydras, right? Because they do have risks out there that regeneration can't fix. But if you look and ask, well, okay, for these ones, at least if you kept them safe, what makes them immortal? It's the fact, I think, that they have very simple anatomy. They have abundant stem cells all through their body, and they're constantly rebuilding their body all the time. Okay, so that's invertebrates. We're not invertebrates. We're vertebrates. Are we different? Are we special? That's where we're going to go next. So before we really get into immortality and regeneration in vertebrates, I want to talk a little bit about lifespan more generally. And we're going to focus on mammals because obviously we are mammals. So here's some things to think about. So first of all, I want to point out just how mind-blowingly variable in size mammals are. One of the smallest mammals is this Etruscan shrew. It weighs in at 1.8 grams. That is really just amazing. That's like 2 mils of water, very little. And it lives for about 2 years. On the other end of the spectrum, as you can see here with this elephant, this tiny elephant to scale, is a large baleen whale, like, for example, the bowhead whale, which weighs, if you want to convert its weight into grams, 100 million grams. So roughly 10 or 20 million times bigger than an Etruscan shrew. Still mammals, though. Maybe not surprisingly to you, whales live a lot longer than mice. So there seems to be some relationship. Smaller animals typically die younger. Large animals, elephants, whales, even humans, we tend to live quite a bit longer. So what does that mean? Well, let's look at this more generally across all animals. So what we're seeing here in this plot is a plot of the mass, the natural log of the mass of the mammals that are in this set, which is a lot of mammals gathered by Speakman here. And then on the y-axis, we're plotting for each one of those mammals, we're plotting their typical maximum lifespan, okay? And so what you can see is this very strong association, whereas animals get bigger, there's a tendency for their lifespan to get longer. That is, you go this way on the chart, you also go up, okay? So most of the points lie along this line here that suggests that big animals tend to live longer than little animals, and little animals, conversely, don't tend to live very long. But you may be noticing something a little funny about this. There's one part of the graph where it doesn't seem to hold up at all. Okay, and there's the shrew and the whale, sorry, for reference. There's this one part of the graph here that doesn't hold up well. And I'm going to get back to Aristotle in a second. So we're going to talk about these animals and who they are in a second. Just want to make the point, right? Aristotle was one of the first people to notice this idea that small animals that have a lot of babies, and that tends to be things like shrews and mice and rabbits, they don't tend to live as long. And so he said it in a more humorous way: salacious animals abounding in seed age quickly. So there's this general trend, but these animals here in the box, they're not matching this trend. Who are these animals? They are bats. Bats, bats. Bats are apparently quite strange. So my colleague, another colleague at the University of Maryland, Jerry Wilkinson here, has been working on bats since he was a graduate student some time ago. And he, in fact, in a recent paper with one of his grad students, Jason Munshi South, points out just how weird bats are. So here's the body mass versus longevity data points for bats as a group. Here are all the bats that they could get data for. Here's every other mammal that we know of. So you can see that bats are just not typical of mammals. There's something weird about them. So what is weird about them? How do they manage to do this? If a bat has a mass typical of a mouse, but lives for, say, 20 years as opposed to 2 years, that's remarkable. How do they do it? And why do they do it? Really, why would there be such variation? Why wouldn't every organism want to live as long as it could? Right? Why would there be short-lived and long-lived animals with the same mass? This is kind of where we're going to go next. So one idea that has a long history in this field is what's called the rate of living theory. And here the idea is that there's something about the actual act of being alive, the metabolism that you have to do to be an organism, that creates damage. And that damage is unavoidable. It might be mutations in your DNA. It might be reactive oxygen species from mitochondria that are damaging your proteins and nerves. There's been a lot of ideas over the years about what it is about living that is intrinsically bad for your body. But this is the idea. And you can summarize this by saying, if you live fast with high metabolism and lots of activity, you tend to die young. On the other hand, if you live very slowly, like say a sea turtle, cold-blooded, doesn't move very much, they live a long time. Okay, so this is the idea. Is this sufficient to sort of explain bats? That is, do bats maybe have really slow metabolism for their size? That would be one possible explanation for why they live so long. So this has been examined, and the paper that I'm going to show you a piece of here is now 30 years old, Austad and Fisher. They aggregated, again, body mass versus, now this time instead of lifespan, we're looking at metabolic rate per gram of animal. Okay, so I want to make sure you understand what we're looking at here. As we go on the x-axis to the right, we're looking at animals that are getting bigger and bigger, and it's a log scale, notice that. As we go up here, we're measuring how much metabolism a given amount of the animal does. So say per gram, how much metabolic activity does that animal have per gram of its body? So this then is no longer looking at body size per se, it's looking at the metabolic rate as a function of a constant piece of that animal. And so what you see then is, like I sort of said in the previous slide, animals that have really high metabolic rate per gram of body tend to be small. And those that have low metabolic rates per gram of their body tend to be quite large. And this runs out of room here at 10 to the fourth grams, but it could go on for several more orders of magnitude. You would say the same exact relationship. So as animals get bigger, they're more and more efficient in terms of their metabolism. They're using less and less energy per gram of their body. And that's probably good because they would be eating way, way, way too much otherwise. Okay, so that's the general pattern for mammals that goes down as you get bigger. And you see that they're pretty tightly clustered around this central line. But if you look at bats within this, and the bats are the markers that are square, you see that they have a slightly lower mean, or not mean, regression line, or expected metabolic rate given their size. So let me put this in another way. Bats on average tend to have a slightly lower metabolic rate per gram of their body than other mammals on average do. But is this enough to explain the difference in lifespan? Well, no, it's not, because the difference in metabolic rate is nowhere near the difference in lifespan between, say, a mouse and a bat. So although there's a little indication that maybe metabolism has a small bit to do with this, it's not sufficient to explain what's going on. So we don't really yet know why bats live longer, but we can think about some more of the reasons why, if there is a way to live longer, they would do it as opposed to other animals. And that gets us to this other major theory of aging biology, which has been referred to as the disposable soma theory. And this was popularized by a guy named Thomas Kirkwood in 1977. And so Kirkwood pointed out that if it costs some kind of energy or effort to maintain your body over time, then it doesn't make sense to put your energy into maintaining your body if you really have no chance of living very long into the future. That is, if your extrinsic risk of death, that is the chance that you're going to be eaten by a predator or frozen to death in the winter, if the chance of that is pretty high, then actually rather than investing in keeping your body going, it would be better off putting that into making babies. Okay. And so the prediction here would be then, well, if you have few predators and maybe not too much seasonal stress, things that would typically kill you, maybe you have a long life. And actually, that fits the bat situation pretty well. If you look at bats, they tend to cluster in caves, right? And other small, hard to get to places up on the ceiling. There's not many things that eat bats. And if you look at them, this is the Mexican free-tailed bat photographed in Texas. Here's an actual video of Mexican free-tailed bats coming out of a cave near. I'm actually not sure where in Texas this is, but you can see they're very prolific. Hopefully nobody's freaking out. So this way of life, flying, makes you less vulnerable to predators. Living in a cave up on the ceiling makes you less vulnerable to predators. And your sheer numbers make you less vulnerable to predators. So this is a reasonable part of the story for why bats live so long, is because they can, right? If they don't die of old age, nothing else is probably going to kill them, and they have a chance to make more babies year after year after year. And in fact, bats do remain fertile into their teens sometimes, whereas a mouse usually is done by age 3. Okay, so now the Kirkwood theory of the disposable soma gives us an idea of why it would be worthwhile for long-lived animals to live long. But it doesn't really tell us how you get there, right? And this is another fascinating question. What is it about the organism itself, the way it works, the way its cells are built, that makes some organisms age faster than others? Why is your dog lucky to make it to 15 when most of us, if we're fortunate, can expect to live into our 70s or 80s? This is a good question, and it's still somewhat a mystery. So I'm going to go back to our single-celled organisms here, remember, and you can actually take cells out of animals and grow them in culture dishes now to kind of treat them as if they were single-celled organisms. This is called cell culture. It's used a lot in biomedical research, and it's been used in aging research in a really interesting way. So you could take fibroblasts, which are cells that would be in your skin, and grow them in dishes. And you can keep track as you grow them how often they divide. So a fibroblast cell in a dish divides once, you've got 2, divides again. And each one of these transfers that involves cell division is called a passage. Okay, so you can count how many cell divisions and passages your cells go through before they kind of crap out and die. And I just want to point that out, that they do eventually die. Cells that are not cancerous cannot be cultured permanently in the lab, but you can culture them for quite a while. And so back in 1991, Roma did a really interesting experiment where he took fibroblasts from a number of different mammals that varied in their lifespan and counted how many of these doublings he could get the fibroblasts to do before they stopped. And so what you're looking at here on the x-axis is the maximum number of cell divisions he managed to get out of these cultured cells from all these different mammals, and you can see it varies over an order of magnitude. And then he plotted against that the lifespan of the whole intact animal. And what I think is really remarkable about this experiment is that he finds that there's a very strong predictive correlation. That is, cells that will go in culture longer tend to be from longer lived animals. And conversely, mouse fibroblasts, remember mice don't live more than 2 or 3 years, they cannot be passaged as many times as human cells can. So it looks then that lifespan is actually a property of single cells of the organism. We don't have to explain it in terms of the organs and the physiology of the whole. The individual cells themselves have different lifespans. And this is really remarkable, but it also suggests that it might be some measurable features of cells that we could get our hands on to try to understand this. So that's where we're going to go next. What is it about cells that allows them to have these variable lifespans? Why do they have variable lifespans? So people have been thinking about this for a while. They presented a number of ideas. And one idea is that, well, animal cells are all full of chromosomes that are linear, not circular. Linear chromosomes have to have their ends protected by these special repetitive sequences called telomeres. And telomeres are needed to prevent the decay of the DNA every time it's replicated. But despite that function, they themselves are gradually lost over the lifespan of the cell. So as you go through that passage kind of experiment that I was telling you back in the previous slide, the telomeres, these purple things, they get shorter over time. And eventually they get so short that the chromosome becomes unstable and the cell commits a kind of suicide called apoptosis. Okay, so you could imagine that maybe telomeres are part of how long-lived organisms live. They must be keeping their telomeres longer. And indeed, that seems to be the case. So there was a paper that came out just last year that looked at this, and they found that in all animals, as we know, telomeres get shorter, like this diagram shows here. And you can measure how much shorter the telomeres get every year. And so if you put that number down here, dots on the right side here then would be mammals where they lose a lot of telomere every year. And over here would be animals that lose a lot less of their telomere every year. So fast loss. And if you plot that against lifespan, sure enough, it holds up, right? The animals that lose their telomeres quickly tend to have small lifespans. The ones that are down here that lose their telomeres slowly tend to have long lifespans. And it's really a remarkably tight correlation. So maintaining telomeres may be one of those things that costs effort, and it may be one of the reasons why short-lived animals live short. They eventually run out of cells that have telomeres. That's one possible explanation for lifespan variation. Another one that has been recently discovered is that there's a change to the base itself. So you remember, hopefully from some biology you had at some point in your life, that the DNA has 4 bases, the A's, the C's, the G's, and the T's. The C is called cytosine, and the base that sticks on the ribose sugar here is this ring. And there's an enzyme called DNA methyltransferase, DNMT, that can put an extra methyl group or CH3 group on that cytosine base. So see this red group here? That methyl group is not usually there, but it can be put there. So this process then of adding these methyl groups to cytosine bases is called DNA methylation. Now, it had been noted for some time that DNA methylation varies with respect to the age of an organism and also with respect to whether a gene that DNA is part of, if the gene is active, it tends to have less methylation. If it's inactive, it tends to have more methylation. So there's a lot of interest in methylation just as a way of regulating the genome and gene activity. So this in general is how we think this works. A gene that is unmethylated is generally accessible and open for business. A gene that has been methylated somehow ends up having its chromatin sort of closed up. And I'm realizing this in this diagram, which I got out of a respected journal, they've kind of got this upside down. Really, the closed heterochromatin is associated with methylation. And the open euchromatin, it should be up here. So they flipped it. I just noticed that. Anyway, you're with me. Closed, tight, inaccessible, gene shut off. Open euchromatin is this state up here with less methylation. Okay, so methylation varies in your genome. You have genes that are shut off in certain cells and they're often methylated. You have genes that are turned on and they're not methylated. And methylation increases with age. So regardless of what gene you're talking about, there's a higher chance it will be methylated as you get older. And we're not the only organisms that do this. It turns out this is sort of an animal thing. So if you, in fact, it's so effective that once you know the sites that are likely to get methylated, somebody can give you a list of how much methylation your different genes have, and you can predict how old a mouse is. So here is an example. These are situations where they've taken mice and they've used this methylation indicator to calculate their methylation age. That's what age subscript met means. It's the predicted age based on methylation. And then there's the actual age of the mouse here. And when you plot those two, they're remarkably correlated. So aging seems to be accompanied by a very predictable gain in methylation at many sites in the genome. This is true for mice, and my colleague Jerry Wilkinson just published a really cool paper that shows that it's true in bats. This is one of the many species they looked at. These are vampire bats. And you can see again, just like the mouse, as the mouse gets older, and these are months, its methylation goes up. And you can predict the age from the methylation itself. So mice and bats both get more methylation as they age. And I was corresponding with Jerry today, and he said that it's his understanding that not only do they both get more methylation with age, but vampire bats, they get methylated at a lesser rate than mice. So it is plausible that the methylation clock, if you will, is ticking more slowly in bats. And so it's not so much that they're big, they're not, they're small. It's not that their metabolism is terribly slow, it's not. But maybe they're maintaining their telomeres and they're holding off methylation better than other animals their size. So that's two likely things that contribute to the extreme longevity of bats. Okay, so I wanted now to say that we have a situation with bats where they live longer than they should, but they still die. They still die predictably. And if you were to, wound a bat, it's not like a planarian. Just because it lives a bit longer than a typical mammal for its size doesn't mean that it has remarkable powers of regeneration. If you run over it with a truck, it's dead, right? So are there animals that not only live an unusually long time, are there some that are good at this regeneration thing? That is, are there any animals that are sort of planarian-like? And so we're going to look at a few that I would argue are getting pretty close, and you might not have heard these before. So obviously, to start with, we humans are not so good at regenerating. This is an unfortunate man who, a long time ago, was, I think, literally run over by a train and lost 3 of his 4 limbs. And you can see that except for healing the stumps, nothing. This is not a salamander, right? They're not growing back. But like I said, salamanders can grow them back. And so here's a situation. This is a newt that has had either a very proximal sort of mid-humerus amputation or a distal amputation, which would be like sort of the middle of the forearm. And either way, this little newt will grow back its complete limb, including its fingers. Now, we can't do that, and most tetrapod animals can't do that. So this leads to a conclusion that I think is pretty safe. Salamanders are badass. They are. They can regrow their limbs. Okay, so another really amazing regenerator is this really cute rodent. And indulge me here a little bit. So this is called the spiny mouse. It lives in Africa. I think this one was taken in Kenya. And they are amazing regenerators. And so I want to wax poetic for a while about spiny mice. So enjoy this cheesy video about spiny mice. And my goal here is to, wait, okay, I got to get out of that and into arrow.
All day long I can hear people talking out loud. When you hold me near, you drown out the crowd. Try as they may, they can never define what's being said between your heart and mine. The smile on your face lets me know that you need me. There's a truth in your eyes saying you'll never leave me. The touch on your hand says you'll catch me wherever I fall. You say it best when you say nothing at all.
They're so cute. Who doesn't want a spiny mouse? They're awesome. Now, if you have a spiny mouse, you might be tempted, because he's so cute, to pick him up like this. This is the way we often pick up mice, lab mice, mice that you have as pets. You pick them up by the scruff of the neck. Oh, such a cute little mouse. That would be a great thing, right? No, don't do that to a spiny mouse. And the reason you should not do that to a spiny mouse, brace yourself, is because this is what will happen. Their scruff will fall off. That's right. If you pick up a spiny mouse by the scruff of its neck, or if you're a predator and you try to eat the spiny mouse by catching it by the scruff of its neck, its skin falls off. It flays itself. It's true. But amazingly, this is not suicide. Because it scabs over and it grows back. It grows back, notice, not as the ugly, hairless, sweat gland free scar tissue that you would have to be stuck with if you had lost that much of your skin. No, it grows back with beautiful fur. And here's an animal in the lab that 30 days later, you can't even really tell that that spot was wounded. This is a wild animal that was caught in Africa, and you can see that this happens to them in nature. And again, they regenerate. So, wow, okay, that's amazing. We can't do that. Scar tissue for us is a very different kind of thing. It's got no hair, it's got no, it doesn't have sensation, it's not really good skin. Um, Part of the reason that this happens is because the skin has very different mechanical properties. And so part of this really cool paper from Seifert et al. In 2012 measures the mechanical properties of spiny mouse skin. And I'm going to walk you through some data here, and hopefully you can follow it. So what we're looking at here is, this is a classic in biophysics. It's called a stress strain curve. And so what we're doing here is we're applying stretch on the x-axis here. And strain is a measure of proportional extension. So for example, 0.4 means that the piece of skin you started stretching is now 1.4 times bigger than it was before. And stress is the amount of physical force being applied to that. So as you add physical force, the skin tends to stretch, right? And so for mouse skin, you can see it's very stretchy. You add stress and it strains quite a ways, right? It can almost be doubled in length before it snaps. But with the spiny mouse, which is in red and black here, you strain them 10% and that's it, they snap and the skin comes off. So clearly this is an adaptation to getting away from predators. They have skin that easily snaps. And that in combination with the ability to regenerate gives them sort of a secret weapon. A predator tries to eat them and instead they just get a little bit of mouthful of fur and that's all they get. So really remarkable. And this has caused a lot of excitement in the 9 years since this paper came out because nobody ever saw this coming. They can also regenerate cartilage in their ear. So it's another unique feature. Okay. So with that tour of immortality in both invertebrates and vertebrates, I thought I might speculate a bit about what might be possible for humans. And this is maybe just the beginning. This is a real growth field in biology. It's not exactly what I work on, but you can see that there's a lot of interest if you look at the literature. A lot of people are researching this and trying to figure out what aspects of the biology of these invertebrate and vertebrate animals that regenerate so well or have long lifespans, what can we apply to us? So the first one is sort of a thing where we don't really have to do anything, but we could maybe measure it and see if it's happening. Oh wait, first of all, yeah, sorry, yeah, let me, before I get into that, let me just start with a reality check. None of what I told you here would suggest that humans will ever be invulnerable to trauma. Explosions, gunshots, knives, like stuff that stops your physiological organism from functioning. You bleed out or you're blown to bits. I don't see any plausible scientific basis for ever being able to survive that. We are not planarians, and even a planarian, you know, if you put it in the barbecue, it's done, right? So we'll always be vulnerable to trauma. But could we use some of these techniques to live longer or be healthier longer? Maybe. So the first one that I want to point out is this idea that maybe we don't have to do anything, we just have to keep living the modern lifestyle that we live. So you can imagine in industrial countries, people are living longer in part because we're taking away those extrinsic risks of death, infectious disease, mass warfare, work accidents. I mean, people still die of all kinds of tragic reasons, right? But the overall rate of mortality due to sort of things outside of your biological control is lower now than it's ever been in history. This then, according to the Kirkwood model, might predict that if we can live longer, predictably, then maybe aging will start to be delayed. That is, by intervening technologically to make our lives longer, it may eventually lead to a biological change in our lifespan. It's an interesting idea. And I thought a piece of that might be worth pointing out also has to do with fertility. Many women in industrial countries are delaying reproduction until much older years than their grandmothers, say, would have. This has been discussed as a possible selection to select for women who can reproduce at older and older ages, which might be expected over, say, thousands of generations, to lead to a situation where women have menopause at a later age. And to give you a sense of how that might work, here's current data that just came out last year of the age at which menopause began in a large set of women from around the United States. And this is just one of the subgroups they looked at. But what you can see is that menopause starts as early as 42, and in some women doesn't happen until 52. So in that 11-year period, there's a lot of variation. As women start to have kids in this sort of higher end of this bracket, there's going to be more and more of us in the country that are descended from women who were fertile at 50. And that will potentially lead to this whole distribution shifting towards the older end. And we may start to see 55-year-old, 60-year-old women more and more routinely. How fast will this occur? Maybe hundreds of years in the future, but it's a reasonable likely thing if we manage to hold our industrial society together and not fall into barbarism. Okay, so that's one idea. Admittedly, very speculative, but you gotta go there a little bit, right? Another one that people are trying to do is to figure out whether they can directly transfer some of what we've learned about the biology of really well regenerating animals to humans. And so remember, I mentioned that salamanders can regenerate their arms. Here's a situation where we've amputated the limb of a salamander, but at the same time, this yellow dot means that salamander had the main nerve that would normally carry nervous signals into the limb. It's been cut. It's been known for a long time that regeneration depends on the presence of nerves where it is possible. And so if you sever the nerve and then cut the limb does not grow back anymore. But by studying these regenerating limbs for a long time, some scientists were able to discover that there is a particular protein in the nerves. It's called NAG. And NAG is apparently the thing in the nerve that is needed to stimulate growth. And the experiment they did is really beautiful. They did the same thing as they did over here. They severed the nerve, they amputated the limb, but this time they added in, by this technique called electroporation, they added in synthetic NAG protein to the stump. And look at that. Adding that back is enough to make it regenerate again. So that's pretty amazing. One protein seems to be largely responsible for the effect of the nerve on regrowth. So what occurs to you immediately then is, well, do we have this protein? The answer is not a very good version of it. Well, could it make a difference if we added it? So that thought occurred to some other scientists, and I just found this paper today. 5 years ago, it was attempted using this Newt anterior gradient NAG protein and putting it into mice that had been experimentally amputated to see if you could get that Newt protein to help them regenerate. And this is not a super impressive result, but there was stimulated bone growth in this experiment over what you would see in a normal salamander. So there's some hope that maybe with a lot more research, we might eventually be able to find things that we can transfer to humans that can make us able to regrow a limb, for example. One of the biggest funders of research in this area, by the way, is DARPA, the Defense Advanced Research Projects Group. They are very interested in trying to help soldiers who've lost limbs in war regrow their limbs. And they're dead serious about finding a way to do this, and they're putting their money where their idea is. So this is an area of ongoing research and really, kind of science fiction, but we may get there. Okay, so I'm going to end. I'm about 48 minutes into this. I just wanted to thank some people that have, over the years, helped me think about biology. My colleagues at the University of Maryland, particularly, I mentioned two of their work, Alexa Bailey and Jerry Wilkinson. My own lab's research is funded by the National Science Foundation. I'm grateful to them for their curiosity and willingness to support the stuff we do. I want to thank the organizers at Balticon, Lisa, and other people that have made it possible for me to be part of this last year and now this year. And I want to thank you all for taking your time on a Friday evening to learn about weird regenerative biology. And with that, I guess I will end my slideshow and turn it back over to Carolyn, the tech host, and see if she wants to help us maybe answer some questions. We've got some time. I'd be happy to take some.
No, you're out of time.
Oh, I'm out of time. Okay.
So thank you very much.
My pleasure. I really enjoyed it, and I will try to catch up with people on Discord or whatever other methods are being used this year.
Homeland, the Homeland place within Discord.
Okay, sounds good. All right, thanks very much.